Here's what the Krebs cycle would look like in a typical science book. The Krebs cycle is located within the mitochondrial matrix, or the open spaces of the mitochondria. It is also called the citric acid cycle or the TCA cycle, and it completes glucose oxidation by breaking down a pyruvic acid derivative, in this case acetyl-CoA, into carbon dioxide. This is a circular reaction because the reaction is ongoing and never reaches the end point. Now notice that this right here is where we would have that transition reaction from the previous slide. And so we would have our 2,3-carbon pyruvic acids from glycolysis coming in, and we would release a carbon from each pyruvic acid, joining with the oxygen that we breathe in to make carbon dioxide. Now that leaves a 2-carbon compound called acetyl that joins with our coenzyme. The coenzyme A helps the acetyl to enter into the cycle. And as it enters into the cycle, the acetyl is 2 carbons. The last structure that is made in the cycle is called oxyacetate, and that is a 4-carbon compound. So when this 4-carbon compound joins with our 2-carbon acetyl, the 4 plus 2 makes a 6-carbon compound called citric acid. And this is the reason that the Krebs cycle is also known as the citric acid cycle, because the first component made in the cycle is citric acid. This is also a picture of the Krebs cycle, but simplified down and to summarize the key steps that occur in the Krebs cycle. So again, right here we have that transitional reaction where we have our 2 3-carbon pyruvic acids, each one releasing a carbon. The carbon joins with the oxygen we breathe in to release 2 CO2 molecules. We go from a 3-carbon compound, losing one of the carbons, to a 2-carbon compound called acetyl. The acetyl connects with a coenzyme. The coenzyme's role is to help the acetyl to enter the Krebs cycle. The coenzyme releases the acetyl and then returns back to pick up additional acetyl molecules, so it is reusable. The acetyl, as it enters the Krebs cycle, joins with the last component that was made in the cycle, known as oxyacetate. This is a 4-carbon compound. That 4 carbons plus the 2 carbons makes a new 6-carbon compound called citric acid. Now, because we have 2 pyruvic acids making 2 acetyl-CoAs per glucose, we require 2 turns of the Krebs cycle per glucose. Each turn releases 2 CO2 molecules, 3 NADH, 1 ATP, and 1 FADH2. Now notice that the NADH and the FADH2 are coenzymes that accept hydrogens and electrons. The NADH accepts 2 electrons and 1 hydrogen, while the FADH accepts 2 electrons and 2 hydrogens. Now, these hydrogens and electrons that are picked up by these 2 enzymes will be used in the 3rd step of cell respiration. And so those hydrogens and electrons here, here, and here will be taken to the cristae of the mitochondria, or the folds, in order for reaction step 3 to occur. Now here we have the 3rd step of cell respiration, known as oxidative phosphorylation. It accounts for most ATP production during cell respiration, averaging between 32 and 34 ATP molecules per glucose. It includes the electron transport chain that is made up of the electron carrier molecules NAD and FADH that are built into the inner mitochondrial membrane. It also uses oxygen that we breathe in to pull those energized electrons from our NAD and our FADH that were harvested during glycolysis, the Krebs cycle, and the transition reaction down the electron transport chain to a lower energy state. Now this is an exergonic slide of electrons, and it's coupled to ATP synthesis. The electron transport accepts the electrons from glucose, from glycolysis, from the transition reaction, and from the Krebs cycle. And it's usually the NAD that carries these electrons, as we saw in those previous slides. However, we saw the FADH picking up hydrogens and electrons in the Krebs cycle. So the electrons are then dropped off, and they're dropped off with a supply of energy, and that energy is needed to pump the hydrogens across from the matrix area to the inner membrane space of the mitochondria. And it's this pumping of these hydrogens, also called protons, that is known as chemiosmosis. Once the NAD and the FAD have dropped off their hydrogens and electrons, they leave and return to the glycolysis, transition reaction, and Krebs cycle to pick up additional hydrogens and electrons. So these coenzymes are both reusable. And when the NADH delivers its electrons, it has enough energy to make 3 ATP per NADH structure. The FADH2 delivers electrons, and it has enough energy to make 2 ATP per FADH2. The cytochrome is the molecule that helps these electron transport system to perform its function. The chemiosmosis is the pumping of the hydrogen across the membrane, and the hydrogen is also known as the protons. And as the hydrogens are pumped across the membrane, we get a large supply of oxygen collected, and that is known as an energy gradient or a hydrogen gradient. And this is going to be used to drive the formation of ATP. The oxidative phosphorylation refers to the production of the ATP. And remember, ATP is formed when ADP and phosphate rejoin. And this is a result of the hydrogen or the protons being moved down the ATP synthase with enough energy to force the ADP and the phosphate back together. And as a result of that, our hydrogens are now on this side of the gradient, and they join with the oxygen that we breathe in. Now, only a small amount of the oxygen we breathe in was attached to the carbon to make CO2 in the transition reaction. The remainder of the oxygen joins with our hydrogen to make water, and that is the reason that 70% of our body is made up of water. So to recap, cell respiration begins with a 6-carbon structure called glucose, and it occurs in the cytoplasm or the cytosol. The first step of cell respiration is glycolysis. 2 ATP are needed to start the reaction. 4 ATP are made with a net gain of 2. We have 2 NADH picking up our hydrogens and our electrons. We end up at the end of glycolysis making a 3-carbon sugar called pyruvic acid. If no oxygen is present, we will go through lactic acid and we will have sore muscles, or we will have fermentation occurring. If oxygen is present, then we're going to go to the Krebs cycle. Now, going to the Krebs cycle, we have our 3 carbons losing a carbon each, and that carbon joins with the oxygen we breathe in to make CO2. And so we go from our 3-carbon compound of pyruvic acid to 2-carbon compounds called acetyl. As a result of that reaction, we have our NADH picking up hydrogens and electrons, and we have a coenzyme helping our acetyl to enter the Krebs cycle. Our 2-carbon acetyl joins with our 4-carbon oxyacetate to make a 6-carbon citrate. We have 4 CO2, 2 ATP, 2 FADH, and 8 NADH at this point. We then enter the electron transport phosphorylation. At this point, we have 32 to 34 ATP made. From beginning to end, we have a net gain of 36 to 38 ATP molecules made per glucose. To put that in perspective, the energy that is yielded in aerobic respiration, going from glycolysis Krebs cycle to oxidative phosphorylation, one glucose can generate up to 38 ATP molecules. If we multiply that 38 times 12 kilocalories and divide that by 686 kilocalories, we end up calculating that cell respiration is 66% efficient. That's about 20 times more efficient than glycolysis alone. Also, consider this compared to a car engine. A car engine is only 25% efficient on our best models. And if we add in the fact that on an average person can live about 30 to 40 days without food. They can live about 3 to 4 days without water. They can live about 3 to 4 minutes without oxygen. They could only live about 3 to 4 seconds without ATP energy. The ATP energy formation must be highly efficient. Lastly, I want to compare photosynthesis to cell respiration. Now we will go over the steps of photosynthesis in an additional video. But the final products of plant photosynthesis, which is the sugar glucose and oxygen, are used by the animal as the beginning substances for cell respiration. The products of respiration are carbon dioxide and water, which the plant needs for photosynthesis. So since photosynthesis and cell respiration are opposite processes, and we can see here this cyclic component, they form a cycle that keeps the levels of our carbon dioxide and our oxygen fairly constant in the atmosphere, which keeps the atmosphere in a livable form for both plants and animals.